imagining then that, okay, so, I don't know, QA or whatever it is, I don't know all my little Z scheme and things. So it's accepted an electron and it gets bigger, but it also then has this, I guess it makes sense, but...
And it changes sort of charge position. So when you're talking of an active enzyme, the possibility that it's then got a portion that can do something like cleave an atom off a molecule.
Or split water, for example.
Yeah, where you can bend this atom into this part.
Ken, we should say, is a medical doctor.
Well, but he will very well explain what is happening. If you have the enzyme has, for instance, let's say that you have the situation that two molecules have to get together in order to produce some kind of chemical reaction. Right? So the enzyme have the ability of actually locking for a while those two molecules, because the enzyme might have the pocket defined by the shape of the whole molecules that will trap one and trap the other, will allow them to interact for a while and will allow them, for instance, to move electron from one molecule to the other molecule. So from the engineering point of view, it's this mechanistic type of the interaction. You have a bunch of atoms. The configuration of the whole molecule is defined, how they are arranged. The electrostatic change on particular atoms will define the shape. But enzymes do actually operate.
Yeah, I mean, I'm thinking about glutarines and
catalyzers. The whole business of the chemical industry, it's similar situation. They have the ability to actually trap the molecules for a while, allow them to exchange the electrons and produce the new kind of the chemical bonds between them. Right? The issue is whether you can get those molecules together and let them stay enough for long enough time that this reaction can happen. So similar thing here is in this business of these electron carriers, right? You get the electron on one of the molecules, take the electron from the water, and in terms of the photosynthetic organism, is this first so-called QA molecule. It has to shift this electron relatively quickly to the next molecule, because if it doesn't, the electron will go back, and the whole thing will go in the path. Nothing is happening. So it is the business how much this reduced form of the molecule, which takes slightly different shape than the oxidized form, will interact with the next one, allowing the next one to take the electron. The question whether the next one will take or not take is defined by the so-called electronegative or electropositive potential, right? And then it goes from one molecule to another, to another, to another.
So I understand this notion that it might bottleneck or slow down. This is what tau is, right?
Yes. So what we are measuring is actually how much time it takes to move the electron from one molecule to the other. So if you plug the pH into this business, pH is the protons in the environment, right? That will screen the negative charges on the surface of the molecule. And if they do so, the molecule can change the shape slightly. And it may happen that the ability of moving the electrons will be different under condition of different pH. Now, the pH is one of the most important factors that controls your physiology. Whether you will need to breathe more or less is defined by the pH in your blood. And again, it is defined only by the fact that this proton screening of some of the molecule will send a signal through some hormones on the other things that will tell that you need more oxygen, because your pH is such that some of the molecules get screwed up and cannot operate properly, right? Medically speaking, is that correct?
No. Somewhere in there.
Okay, that's good enough.
Yeah, no, I get it. So I would have asked anyone this.
Why don't you ask a lot about how common is hemoglobin and chlorophyll?
No. I want to ask my question, and then you can ask your question, all right?
You're going to ask that one.
Can electrons in the Z scheme, move backwards accidentally, or is there a case where you don't? Because I used to think it was unidirectional, or is it unidirectional exclusively?
All enzyme systems have leak back.
They always can move back, yes.
Yeah, where it depends on the drivers each side. Where if you change the concentration of things, if you have lots of precursor, no product, and you have the right pH and conditions,
And it should drive forward.
you have lots of product and almost no precursor, it'll tend to go back the other way. It's an equilibrium. You can push it one way or the other by adjusting effectively the pressures.
It's dangerous if you've separated energy and you've gone halfway in your reaction and then you come back, you've got a whole bunch of energy you've got to get rid of. And so that's what happens to photosystems. If you go backwards, you've got energy that you've got to get rid of, and that's reactive oxygen.
And not necessarily, the pathway this way might work, but the enzyme isn't necessarily made for doing the opposite, so it won't necessarily go, can't do that, okay, I'll take the electron back, take the oxygen, and you go back there. It's more likely to go, we'll oxidize the enzyme, and now the enzyme doesn't work.
Cause more damage.
So one thing we are discussing right now, we have this phenomenon, this business of that we cannot explain for many years, and many bottles of wine. That is actually related to the fact that if you produce the charge separation in the photosynthesis, right? You take electron from the water, you put this electron in the so-called electron acceptor, but you take this electron from one molecule that for a period of time has positive charge because obviously the balance is then the question is how quickly you can move this electron forward so it cannot back react with this positive charge, and how quickly this positive charge on the donor side can take electron from the water so it's no longer is susceptible to this back reaction. And we think we can monitor the intensity of this back reaction because one of the parameters that we measure we think, or I think, or I stupidly think, is related to this back reaction, that there is some situation when the electron on this donor side that's supposed to go forward and end up in the photosystem 1 and actually back react with the positive charge in the donor side, and during this back reaction produce the signal, the radiative signal that we observe as the fluorescence. So, as he said, it's always the equilibrium between the rate of going one way and the rate of going the other way. And if there is another process that can steal this electron, it will happen. The issue is to establish all those rates that you have relatively efficient transfer of this excitation energy in the direction you would like to have.
And an enzyme can only catalyze a reaction. That reaction will go ahead anyway, depending on the energy levels of things. It just needs to go over a higher mountain. The enzyme lets you go around the side and not have to go to the same peak energy, which means that a lot of things that are driven by an enzyme, if the enzyme's not there, it still happens at a low level. So something like in the pancreas where you make digestive enzymes, but it's secreted as a proenzyme, they're not active. And they get secreted down the duct into the digestive tract. The acidity of the digestive tract helps activate them, and then they become nasty proteolytic enzymes that chomp up any bit of protein. You block your pancreatic duct. You secrete enzymes, or proenzymes. There's a small rate of them converting into active enzymes. then activate other enzymes and promptly chomp your pancreas up.
Really bad.
So it's not like the absence of one factor actually stops it completely happening. It just may provide a faster rate or a more definite sort of direction.
So thermodynamically it's easier.
But one thing that he mentioned is the acidity. What is this acidity? The amount of protons that can manipulate the shape of the molecules.
Well, molecules change shape dependent on pH, changes the...
But the protons also have positive charges and are presumably then attracting in electrons or to balance their
Well,
charge.
if you've got negative charges on a molecule and you've got this positive proton hanging around, they'll come and associate.
They can
And it-
screen
and
those
it-
negative
and
charges,
it might-
neutralize them,
It
right?
might be like an ionic sort of association, it might be a covalent, it might just be a sort of loose association in the solution.
Yep.
But that all affects how things go.
I'm right. Very interesting. This is pure gold. Lisa doesn't have to come to the lab tomorrow.
Okay,
So-
to make it even more pure gold,
Yes.
Another bottle of wine.
I'll go organise that. So, I need a description of the commonality between hemoglobin and chlorophyll.
Yes. I want to know
There you go. Humans and plants have got the same same structural protein as the centre to our oxygen movement system and our oxygen splitting system.
Can you just go into that a little bit more? With you're saying that chlorophyll is
Is of the same group as chlorophyll. Chlorophyll and hemoglobin are both
are structurally similar